Myself, Dr. Mrinmayee, working as an assistant professor in the Department of Anatomy, SNIMS College. Today in this session, we are going to discuss about the development of ear. So we know that the embryology portion of the anatomy is very difficult to understand. So I have tried to make in a very simplified way so that the students can understand the development of ear. So we are going to see under the following heading. First, we will see the different parts of the ear, then the development of the inner ear, middle ear, external ear separately. After understanding of all this development, we will try to correlate clinically, because at the end, all this knowledge we are going to apply clinically. And at the end, we will summarize the topic. So we know that at 4th to 5th week of intrauterine life of the embryo, the pharyngeal arches develops. Now, in this picture, what you're seeing here, these are the pharyngeal arches, and these are the pharyngeal cleft. What you are seeing here, these are the pharyngeal cleft. Now, the first pharyngeal cleft, this is the first pharyngeal cleft, which develops into the external auditory meatus. The first pharyngeal cleft develops into the external auditory meatus. The first pharyngeal arch, so this is the first pharyngeal arch. The first pharyngeal arch develops into the malleus, incus, tensor tympani. Malleus and incus, these are ear ossicles. Apart from this malleus and incus, there is another ear ossicle, that is stapes. While the malleus and incus, they are developing from the first pharyngeal arch, the stapes develops from the second pharyngeal arch. An associated muscle with the stapes, it develops from the second pharyngeal arch. So in this picture, you can see here, these are the pharyngeal arches, first, second, third, fourth, and sixth. As we know, the fifth pharyngeal arch disappears. So, and these are the first, second, third, and fourth pharyngeal cleft. So the first pharyngeal cleft develops into the external auditory meatus. First pharyngeal arch develops into the malleus, incus, and tensor tympani. So tensor tympani is the associated muscle along with these ear ossicles. And from the second pharyngeal arch, it develops into the stapes, that is another ear ossicle, and the muscle associated with stapes, that is stapedius. So the development of any structure, when we read, we have to always correlate with the gross structure. Okay, so it is not like that separately we are going to read all these structures. When we are going to read about the development of ear, first you can read about the development of the ear. Alongside, you can read about the gross structure of the ear. By that only you can understand the topic or the structure very easily. And also, from the first pharyngeal pouch, it develops into the tubotympanic races. Okay, the first pharyngeal pouch, it develops into the tubotympanic races. Now, the part of the ear, we know the ear we can divide into external ear. Okay, so this is the external ear. From outside, what you are seeing, that is the pinna or the auricle, up to the external auditory meatus. Okay, so pinna or auricle and external auditory meatus, these are the parts of the external ear. After that, there is a membrane or curtain or a partition between the external environment and the internal environment of our body. So that is called as the tympanic membrane. So starting from the tympanic membrane, then the malleus, incus, and the stapes, these are the ear ossicles. Then the tympanic cavity and the muscles associated with the ear ossicles, these are tensor tympani, which is associated with the malleus and incus, and the stapedius muscle, which is associated with the stapes. So these structures are related or the component of the middle ear. Now, what are the component of inner ear? Inner ear, the component are the bony labyrinth. This is what you are seeing, that is the inner ear or internal ear. And what is structure, snail like structure, what you see, that is cochlea. So cochlea we can actually compare with the structure of snail. Okay, now it is like a bony shell. And as it is a bony shell, that means it's a part of the bony labyrinth, which covers the membranous labyrinth. So bony labyrinth is the shell or covering or the envelope of the membranous labyrinth. So till now what we have seen, that we know that the external ear, it consists of auricle and external auditory meatus, starting from the tympanic membrane or the eardrum, then the ear ossicles, okay, malleus, incus, and this is stapes, associated muscles, tensor tympani and the stapedius. Along with that, the cavity in the middle ear, that is the tympanic cavity. These are the component of the middle ear. And the inner ear, it consists of bony and membranous labyrinth. Bony labyrinth is the part of the inner ear which covers the membranous labyrinth. Now we will see the development of each component separately. So let us first see the development of inner ear. So internal ear, it develops at the embryo when it is of 22 days old. Okay, so when the embryo is of 22 days old, it is composed of 8 pairs of somites. Okay, so the age of the embryo, how we can determine, or that is by the pairs of the somites it is composed of. Okay, so at the 22nd day or 22 days of age, the embryo, it consists of 8 pairs of somites. At that time, the length of the embryo is around 2 millimeter. So at that time, the length of the embryo is around 2 millimeter. So when the embryo is at 22 days of age and it is composed of the 8 pairs of somites, at that time, 2 primordia, that means from where the internal ear is going to develop, that appears. These 2 primordia are otic vesicle and the mesenchyme surrounding the otic vesicle. Okay, so the otic vesicle and the mesenchyme surrounding the otic vesicle are the primordia of the development of internal or inner ear. Otic vesicle, it develops into the membranous labyrinth, and the mesenchyme surrounding the otic vesicle develops into the bony labyrinth. So as already I have mentioned here, and we have learned till now, that the internal ear or the inner ear, it consists of 2 components, that is membranous labyrinth and bony labyrinth. Membranous labyrinth is covered by the bony labyrinth. And what are the 2 primordials of this internal ear? These are the otic vesicle and the mesenchyme surrounding the otic vesicle. So let us see. Now here in this structure, what you are seeing, that is the neural tube, and over that, what you are seeing, that is the surface ectoderm. So we know that when the any structure we show as the ectoderm, that is colored as blue color. Okay, so by this you can easily remember which is the ectoderm, mesoderm, and endoderm. Now this otic blackboard is an ectodermal thickening. Okay, the otic blackboard is an ectodermal thickening on either side of the developing rhombencephalon. So you can see here, this is the wall of the rhombencephalon. On either side of this rhombencephalon, the ectodermal thickening occurs, that is called as otic blackboard. Okay. Now, what happens? The next step is that you can see this image also, and side by side, the this right-sided image also is the same. So here you can see the otic blackboard. After that, it invaginates into the underlying mesenchyme. So what you see, this is the mesenchyme. So the otic blackboard invaginates into the underlying mesenchyme. By that time, it is called as otic pit. What do you mean by pit? It is any depression, right? So the otic blackboard, it invaginates into the underlying mesenchyme, and now it is called as the otic pit or otic cup. Okay, so here in this image it is mentioned as otic cup, and here it is mentioned as otic pit. Both are same. Okay, so the otic blackboard, it appears as an ectodermal thickening. So it appears as an ectodermal thickening on either side of the developing rhombencephalon. After that, this otic blackboard, it invaginates into the underlying mesenchyme, and it forms into the otic pit, that is a depression on either side of the wall of the rhombencephalon. So this otic pit is also called as otic cup. This otic pit is also called as otic cup. Now, when this depression has formed, what happens now? The two edges of the otic pit, it will fuse together. As the depression, it will become more deeper. Okay, so it will become more deeper. So as a result, what happens? The two edges or the two ends of the otic pit, it fuses together. So as it fuses together and it unite to form the otic vesicle. Now it is called as otic vesicle. So what is the otic vesicle? That is ultimately the primordia of the membranous labyrinth. Okay, so primordia of the membranous labyrinth. So these two edges of the otic pit or otic cup fuses together to form the otic vesicle. So as they have fused together and formed the otic vesicle, so definitely it has been incorporated from the surface ectoderm into the underlying mesenchyme. Now in this structure, you can see here, this is the otic vesicle, correct? Now here also you can see this is the otic vesicle which has been detached from the surface ectoderm. Now, just to remember here, just see the surrounding structure. So an otic vesicle is surrounded by the mesenchyme. Okay, so the otic vesicle is surrounded by the mesenchyme. So at this level, what we have learned till now, so we have learned in this slide that the otic blackboard has been formed as an ectodermal thickening on either side of the developing rhombencephalon. The otic blackboard then invaginates into the underlying mesenchyme and it forms into the otic pit. or it is also called as the otic cup. Now this otic pit, two ends of this pit, it fuses together and it gets detached from the surface ectoderm. Now and it gets detached from the surface ectoderm. It has now formed into the otic vesicle. Okay, the otic vesicle, to remember here, it is surrounded by the mesenchyme. Now, why I'm telling so? To understand the next step of the development. Okay, now this otic vesicle is also called as autocyst. Okay, otic vesicle is also called as autocyst. Now, we have understood the development of this membranous labyrinth. Now let us see the next step. Now this otic vesicle. So till now we have learned the development of the otic vesicle or the formation of the otic vesicle. So the other name of otic vesicle is autocyst. Now in this image you can see this is the otic vesicle, and from the otic vesicle a diverticulum has been developed which is called as endolymphatic duct. Okay, so endolymphatic duct has been developed from the otic vesicle. So as the development or appearance of the endolymphatic duct, otic vesicle now it is divided into two portions. Okay, so it has been divided into two portions. The two portions are ventral saccular part and the dorsal utricular part. Ventral saccular part and the dorsal utricular part. Now the ventral saccular part will develop into the saccule and duct of cochlea. It will develop into the saccule and duct of cochlea. The dorsal utricular part will develop into the utricle and three semicircular duct. Okay, it will develop into the utricle, three semicircular duct. Now, in this image, you can see here, this is the utricle. Now, from the utricle, three disc-like evaginations. Okay, so this yellow color, what you see, these are the three disc-like evaginations which are developed as the ducts. Okay, now what happens? How the ducts are developed when the central portion of this utricle disappears. After that, what happens? The ducts will develop as right angle to each other. So first, the superior semicircular duct will develop, followed by that lateral semicircular duct, and then posterior semicircular duct. Now, in this image, you can see the structure that is the ampullae. Ampullae is nothing but the dilated portion at one end of this semicircular duct. So, you can see here the dilated portions in the one end of the semicircular ducts. Okay. Now, from the saccule develops into the saccular part, it develops into the cochlea duct. So in this image, you can see this pink color, that is the cochlear duct, and it elongates from the saccule. What happens? A tubular diverticulum will develop. After that, it elongates. So in this image, you can see that the cochlear duct has been developed. After that, it will elongate as a tubular diverticulum. It will elongate as a tubular diverticulum. Then it turns spirally. In this image, number F, you can see it turns spirally and it will turn around 2.75 turn. Okay, so it will turn around 2.75 turn. And ultimately it forms the duct of the cochlea. Soon after that, there is a communication which is narrow. Okay, the narrow communication will form between the duct of the cochlea and the saccule. Now, all these structures you have to correlate along with the cross structure. Okay, otherwise it is very difficult to understand. Now here, what we have seen till now. So let us again revise about the membranous labyrinth. So in the membranous labyrinth, we have seen that the primordia of the membranous labyrinth has been formed in this slide, that is the otic vesicle. So what happened? So at first the otic plateaued as an ectodermal thickening on either side of the developing rampant saphlon will formed. Okay. Now from this, the otic plateau will invaginates into the underlying mesenchyme, and at that time it is called as the otic pit or otic cup. Now the two ends of the otic pit, they fuses together and gets detached from the surface ectoderm. As a result, it is forming the otic vesicle, which is also called as otocyst. This otic vesicle is surrounded by the mesenchyme. In this slide, we have learned that the otic vesicle has been formed. From there, the diverticulum endonymphatic duct has been appeared, and this endonymphatic duct appearance, it formed or it gets developed into the otic vesicle into two portions: ventral saccular part and the dorsal utricular part. Dorsal utricular part develop into the utricle and three semicircular duct: superior or anterior semicircular duct, lateral semicircular duct, and posterior semicircular duct. And also the ampullae, that is the dilated portion at one end of the semicircular ducts, has been formed. From the saccular part, that is the ventral part, it develops into the saccule and the duct of cochlea. Now you have to remember the duct of cochlea, it turns around 2.75 turns. Now, as we know that the nerve concerned here, it is vestibulocochlear nerve. So the vestibulocochlear ganglion, how it is formed? So in this picture, what you are seeing, this is the vestibular ganglion. Now this vestibular ganglion is formed from the cells migrated from the otocyst along with the neural crest cells. So along with the neural crest cells, the cells which are migrating from the otocyst, it will form into this vestibular ganglion. Now this vestibular ganglion, it consists of bipolar cell neurons. Okay, so the vestibular ganglion, it consists of bipolar cell neurons. So as it is bipolar cell neurons, that means it will have the peripheral process and central process. Now this peripheral process are innervating the saccule, utricle, semicircular duct. This saccule, utricle, and semicircular ducts, these are conveying or these are responsible for our sense of equilibrium, that means maintaining our balance of the body. Whereas the central processes of this bipolar cell neurons will form the vestibular nerve itself. So we know that the vestibulocochlear nerve is composed of two components, vestibular component and the cochlear component. Vestibular component of the vestibulocochlear nerve deals with the sense of equilibrium of it, maintains the balance of our body. So vestibular ganglion has been formed by the cells which migrates from the otocyst along with the neural crest cells. It consists of the bipolar cell neurons. As it is bipolar cell neurons, it consists of peripheral processes and central processes. Peripheral processes innervates the saccule, utricle, and semicircular ducts, which are responsible for the sense of equilibrium. Central processes, they are forming the vestibular nerve. Now next is the cochlear ganglion. Cochlear ganglion can be also called as spiral ganglion. Why it is called as spiral ganglion? Here you can see the arrangement of the ganglion. You see it. So what happens? The cells it migrates along with the coils of the membranous cochlea. So the cells, as it is migrating along with the coils of the membranous cochlea, so the arrangement is like spirally, as we know that the duct of cochlea has been formed as a tubular diverticulum, and after that it turns spirally. Okay, so the cells are also migrating alongside the coils of this membranous cochlea. Because of that, the cochlear ganglion is also called as spiral ganglion. Now, this cochlear ganglion is also consisting of bipolar cell neurons, as like the vestibular ganglion. So, bipolar cell neurons means it has two processes: peripheral processes and central processes. Peripheral processes of this bipolar cell neurons terminate in the hair cells of organ of Corti, which convey the sense of hearing, and the central processes of this cochlear ganglion of the bipolar cells, it will form into the cochlear nerve. So the cochlear ganglion has been formed as by the cells which migrates along with the coils of the membranous cochlea. Because of that, it is also called as the cochlear ganglion. It consists of bipolar cell neurons. So the peripheral processes and the central processes. Peripheral processes inhabits the hair cells of organ of Corti, which conveys the sense of hearing, as it is now involved in the auditory pathway, and the central processes, they form the cochlear nerve. Now, after learning about the membranous labyrinth, let us see about the bony labyrinth. Now, as if you can remember till now that we have learned the otic vesicle. So here you can see this is the otic vesicle, right? Now the otic vesicle, here this blue color, it is showing the mesenchyme. So we have seen in the previous slides. You can see, yeah. So here you can see this is the otic vesicle or the otocyst, and it is surrounded by the mesenchyme, right? So this is the otic vesicle which is surrounded by the mesenchyme. Okay. So the otic vesicle, it is induced by the cells of this otic vesicle to condense, and it forms into the otic capsule. Okay, now it has been formed into the otic capsule. Okay, now the surrounding this, we can see some spaces. Okay, so the spaces are called as the peri-lymphatic spaces. Now these peri-lymphatic spaces are filled with peri-lymph. These peri-lymphatic spaces are filled with peri-lymph. These spaces has been formed by the dedifferentiation between the membranous and the cartilaginous part. Okay, it has been formed between the cartilaginous and the membranous part, and the spaces are filled with the peri-lymph. Now, these peri-lymphatic spaces which are filled with the peri-lymph, they are communicated or connected to each other at the apex of the cochlea. So, these peri-lymphatic spaces, they are communicating with each other at the apex of the cochlea, which is known as helicodrama. Okay, which is known as helicodrama. So, the apex of the cochlea is known as helicodrama, where the peri-lymphatic spaces are connected with each other. Now, these spaces, this is the Scala vestibuli. Now the scala vestibuli is separated from the cochlear duct, which is also called as the scala media. Okay, so the scala vestibuli, which is consist of perilymph, and the scala media or the cochlear duct, which is consist of endolymph. They are separated from each other by means of vestibular membrane or Richter's membrane. So in this image also you can see this is the vestibular membrane or Richter's membrane. The scala tympani, now the scala tympani, which is filled of perilymph, and the scala media, which is filled of endolymph, are separated from each other by the basilar membrane. So scala vestibuli is separated from the scala media by vestibular membrane, which is also called as the Richter's membrane. Scala tympani is separated from the scala media by the basilar membrane. Okay. Scala vestibuli, it consists of the perilymph. Scala tympani is also consists of perilymph, and the scala media is consist of endolymph. Now, how this cartilaginous otic capsule, after that it ossifies and forms into the cochlea. Okay, it forms into the cochlea, and the modulus of the cochlea, around which the cochlear duct spirally turns, that modulus has been formed by the direct ossification from the mesenchyme. Okay, modulus has been formed by the direct ossification from the mesenchyme. Cartilaginous otic capsule, it ossifies and forms the cochlea. So cochlea means the bony labyrinth, which is the covering of the duct of the cochlea, which is the part of the membranous labyrinth. So at this level, we have seen that the bony labyrinth has been formed, membranous labyrinth has been formed. So in this blue color, you are seeing that is the superior semicircular canal. Then you can see the lateral semicircular canal, and you can see the lateral semicircular canal, and this is the posterior semicircular canal. Okay, so it is enclosing the semicircular duct. It is enclosing the semicircular duct. So here the orange color, it denotes the semicircular duct. And this is vestibule, which encloses the utricle and the saccule. It encloses the utricle and saccule. Together they are called as otolith. Organ. Now this snail-like structure, this is known as the cochlea, which encloses the duct of cochlea. Okay, so it encloses the duct of cochlea. So by this time we have learned the development of internal ear, composed of two parts, membranous labyrinth and the bony labyrinth. Okay. Membranous labyrinth is covered by the bony shell, that is bony labyrinth. What is the part of the bony labyrinth? The semicircular duct, superior semicircular duct, lateral semicircular duct, and posterior semicircular duct. Now, after learning the development of the internal ear, now let us see about the development of middle ear. Now, what are the components of middle ear? Middle ear consists of tympanic cavity, auditory tube, ear ossicles, tympanic antrum, and mastoid ear cells. Tympanic cavity and the auditory tube, how it is formed? It is formed from the first pharyngeal pouch. Okay. Now what happens in this picture? What you're seeing, this is the first pharyngeal pouch, Now this first pharyngeal pouch will develop into a tubotympanic recess. Okay, it developed into a recess which is known as tubotympanic recess. Now the tubotympanic recess, it will develop in a lateral direction. Okay, so it will develop into a lateral direction. As it is directed laterally, as it develops laterally, it comes in contact with the first pharyngeal cleft. That is the surface ectoderm. So the tubotympanic recess, it extends laterally and comes in contact with the, here you can see this is a blue color, that is the first pharyngeal cleft. So it comes in contact with the ectoderm. So what you are seeing here, this is the endoderm. This pink color is the mesoderm, and this blue color is the ectoderm. Okay. Now the tubotympanic recess, the distal widened portion. Okay. So the distal widened portion. Can you see here? This is the distal widened portion. Okay. So the distal widened portion is forming into the tympanic cavity. The distal widened portion is forming into the tympanic cavity. Okay. The narrow proximal portion. Can you see this narrow proximal portion? So this narrow proximal portion will develop into the pharyngotympanic tube, which is the auditory tube, can also be called as the esophageal tube. Okay. So that is about the tympanic cavity and auditory tube. So how it is formed? The tubotympanic recess, it develops as a the extension. Okay, it develops as an extension towards the lateral direction, and it comes in contact with the surface ectoderm. Okay, it comes in contact with the ectoderm, that is the first pharyngeal cleft. So here what you are seeing here, so the tubotympanic recess, when it is going to the lateral direction, the distal widened portion, it develops into the tympanic cavity, and the narrow portion, it develops into the auditory tube. So by this time we have learned about the development of tympanic cavity, auditory tube. Now let us see about the ear ossicles. Now here in this picture you can see the ear ossicles, the malleus, incus and the stapes. Now the malleus and incus, they are formed from the dorsal end of the cartilage of the first pharyngeal arch. So this is the first pharyngeal arch. Okay. Now the dorsal end of this arch, from the cartilaginous part, you can see the malleus and the incus has been formed. The cartilage of the first pharyngeal arch is called as Meckel's cartilage. Okay. It is called as Meckel's cartilage. Now the stapes is formed from the dorsal end of the cartilage of the second pharyngeal arch. The cartilage of second pharyngeal arch is called as Richard's cartilage. It is called as Richard's cartilage. So the malleus and incus has been formed from the dorsal end of the Meckel's cartilage. Stapes has been formed from the dorsal end of the Richard's cartilage. The muscle associated with this ear ossicles, that is tensor tympani, with the malleus and incus, it is developed from the mesenchyme surrounding the cartilage. So as it is developed from the first pharyngeal arch, so it is supplied by the mandibular nerve of the trigeminal ganglion. Okay, so we know that the trigeminal nerve or the trigeminal ganglion has got three components, right? Mandibular component is devoted to this first pharyngeal arch. Now the stapedius muscle, which which is associated with the stapes that is developed from the mesenchyme of the second pharyngeal arch. Thus, how it is supplied by the facial nerve. So we know that the nerve of the first pharyngeal arch is mandibular nerve, and the nerve of the second pharyngeal arch is facial nerve. So the nerve supply of tensor tympani is mandibular nerve, and the nerve supply of stapedius muscle is facial nerve. So, now about the tympanic antrum and the mastoid hair cells. Now the tympanic antrum. Now the tympanic antrum, how it is formed? The tympanic antrum will form during the fetal life by the vacuolization of the cells of this tympanic cavity. Okay, the vacuolization of the surrounding cells of tympanic cavity, it will form into the tympanic antrum. What about the mastoid hair cells? Mastoid hair cells will develop, that is, the full development will completed by the age of 2 years of age. Okay, so when the child is of 2 years of age, by that time only the mastoid hair cells will be developing. And inside that the mastoid process, so you must have learned about the mastoid process, which is a part of the temporal bone. So there only the mastoid hair cells will well developed at the age of 2 years. Okay, so the because of this direct or close contact with the tympanic cavity and the tympanic antrum, what happens? The infection of the middle ear, it spreads so easily to these hair cells. Okay? So till now we have learned about the development of internal ear and development of middle ear. Now let us see about the development of external ear. So external ear, what are the parts of external ear? We have learned that a pinna or auricle, that tympanic membrane, then the external auditory meatus. Okay. So let us see the development of external auditory meatus. External auditory meatus, it is formed as a primitive meatus, then it will ultimately develop into the meatal plaque and then the definitive meatus. Now the primitive meatus, in this image you see here. Yeah. So this is the first pharyngeal flap. Okay, so this first pharyngeal flap, what happens? The first pharyngeal flap, it will invaginate as a funnel shape. Okay, it invaginate as a funnel shape and develop into the primitive meatus. Okay, so primitive meatus has been developed as a funnel shaped invagination from the dorsal part of the first pharyngeal flap. Now, after that, the cells which are present in the floor of the external auditory meatus, okay, which is present in the floor of the external auditory meatus, these epithelial cells will proliferate. The epithelial cells will proliferate and it will form into the meatal plaque. Okay, so it will form into the meatal plaque. So here you can understand that if the proliferation of these cells fails, so it will definitely lead into any abnormality. So the external auditory meatus has been formed as a funnel-shaped invagination from the dorsal part of the first pharyngeal cleft. The epithelial cells which are present in the floor of the first pharyngeal cleft will proliferate and will form into the meatal plaque. Then what happens? Dissolution of this meatal plaque at the age of 7th month of embryo, it will form into the definitive meatus. Okay, so it will form into the definitive meatus. So how the external auditory meatus has been formed? First, the primitive meatus has been formed as a funnel-shaped invagination from the dorsal part of the first pharyngeal cleft, meatal plug will form by the proliferation of epithelial cells which are present in the floor of first pharyngeal cleft. Dissolution of meatal plug will form into the definitive meatus at about 7 months of the embryonic life. Now let us see the development of tympanic membrane. So the tympanic membrane, as I have mentioned already, that it is the partition between the external environment and the internal environment. So the tympanic membrane, if you see in this image, in this ring, you can see. So blue color means the ectoderm, right? So here you can see the disc-shaped or ring-shaped tympanic membrane. So the tympanic membrane, it has got 3 layers. Okay, outer cuticular layer. Okay, outer cuticular layer. Then intermediate fibrous layer and inner mucus layer. Okay, outer cuticular layer is developed from the ectoderm of first pharyngeal pouch. It develops from the ectoderm of first pharyngeal pouch. Inner mucus layer, it develops from the endoderm of tubotympanic recess. Inner mucus layer develops from the endoderm. So you can see the inner mucus layer. Okay, inner mucus layer, it develops from the endoderm of the tubotympanic recess. So till now we have learned that ultimately the tubotympanic recess develops into the middle layer also, and also it contributes to the formation of the part of the external layer, right? So the tubotympanic recess, it develops into the tympanic cavity and auditory tube or the pharyngotympanic tube, and the part of the external layer, that is tympanic membrane, inner mucus layer, is also formed from the tubotympanic recess. So it gives 3 contributions. Now the intermediate fibrous layer, it is formed by the adjoining mesoderm of first and second pharyngeal arches. So it has been formed by the adjoining mesoderm of first and second pharyngeal arches. So the tympanic membrane is a structure which develops from all 3 germ layers. These 3 germ layers are ectoderm, mesoderm, endoderm. Ectodermal layer is the outer cuticular layer which develops from the ectoderm of first pharyngeal pouch. Inner mucus layer develops from the endoderm of tubotympanic recess. So, and the intermediate fibrous layer, it develops from the mesoderm of the adjoining adjoining mesoderm of the first and second pharyngeal arches. So in this image you can see this is the outer cuticular layer, intermediate fibrous layer, and the inner endoderm layer. Okay, so in the most of the images it has been seen that the ectoderm is shown by the blue color, mesoderm is shown by the, most of the time it is by the pink color, and endoderm you can see the orange or the yellow color. So the tympanic membrane, till now we have learned it has got 3 layers. The definitive tympanic membrane formed during the recanalization of external auditory meatus. Prominent ring shape. Then the it will support the tympanic membrane. Okay, so the prominent ring-shaped neural crest derived bone called as the tympanic ring supports the tympanic membrane. Tympanic ring will get absorbed into the temporal bone. Tympanic ring will get absorbed into the temporal bone. So till now, the definitive tympanic membrane has been formed. Now let us see the development of the auricle or pinna, which we are seeing from the outside and which the normal people called as the ear. So that is the auricle or the pinna. So auricle or pinna, it develops at the 5th week of embryo, and and it develops from 6 hillocks. Okay, so these are the 6 hillocks. 1, 2, 3, 4, 5, 6. Now this 6 hillocks from the first and second pharyngeal arch. First pharyngeal arch forms the tragus, crus of helix, and the helix. Okay. Second pharyngeal arch, that is 4th, 5th, and 6th hillocks, they develop into the antihelix, antitragus, here lobule. So in this image, you can see here that the first pharyngeal arch, which is the mandibular arch, it has been shown as a pink color, and the second pharyngeal arch has been shown as a blue color. So the first pharyngeal arch, it will develop into the tragus, crus of helix, and the second pharyngeal arch will develop into the antihelix, antitragus, and the ear lobule. So the helical margin is markedly raised and folded at 12 weeks of embryo. Development of external ear is completed by 16th week of embryonic life. As the mandible develops, the auricle gradually translocates to lateral and to the cranial side. So the development of external ear is completed by the 16th week of embryonic life. As the mandible develops, auricle gradually translocate and to the lateral and the cranial side. So till now we have learned the development of inner ear, then development of middle ear, and development of external ear. After understanding of all this development, let us correlate clinically. It's a congenital deafness. How it occurs? Failure of the conduction of sound from the external ear to cochlea. Okay, failure of the conduction of sound from the external ear to cochlea is the congenital deafness. It occurs due to failure of canalization of meatal plug. So, if you can remember, the meatal plug is formed. After that, the canalization occurs. So, if failure happens, it will lead into the congenital deafness, because at that time the sound will not get transferred from the external ear to the cochlea. Congenital stenosis or atresia of the external auditory meatus, congenital malformation of auditory ossicles and eardrum, absence of the external meatus and tympanic cavity. This all will lead to the congenital deafness. Congenital sensory neural deafness. It occurs due to abnormal development of the membranous labyrinth, failure of the auditory nerve to make contact with developing orthocyst, damage to hearing apparatus by prenatal and perinatal factors. This all will lead to the sensory neural deafness. Now, in this picture, you can see here the pinna is underdeveloped. So this condition is known as microtia. Okay, so this condition is known as microtia. Here the pinna is underdeveloped. It is unilateral, occurs in right ear. Males are most predominant, and risk factor is maternal diabetes mellitus and advanced maternal age. So if microtia is present, so definitely macrotia will be also present. So what is macrotia? Macrotia is the abnormal enlargement of the pinna. Okay, macrotia is the abnormal enlargement of the pinna. It is associated with the, or it is seen in the fragile X syndrome. So these are the chromosomal defects. anotia. In this image, you can see the absence of auricle. Okay, the absence of auricle, it is known as the anotia, and it is associated or seen in first arch syndrome. This is a frequent condition seen as the pre-auricular sinus. So here you can see the sinus. So what is that? The skin tags and the shallow depressions anterior to the pinna. Pre means anterior, right? So anterior to the pinna. This occurs due to abnormal development of the auricular helix. So if any abnormal development occurs in the helix, so it will lead to this pre-auricular sinus. It is quite familiar and seen in patients coming to the ENT OPD, and it is frequently bilateral. Okay, so it is frequently bilateral, and it is unilateral at that time. Commonly, it occurs in the right side. So frequently, it is seen bilaterally, but if it is unilaterally present, so that time the right side is getting affected more. So at the end of the session, we have learned that the ear, three parts: inner ear, middle ear, external ear, otic vesicle, and the otic capsule. Okay, they are the primordia of the internal ear. Okay, development of internal ear. Two primordia are present. These are the otic vesicle and the otic capsule. Okay, otic vesicle and the otic capsule. And after that, what happens? We have seen the tubotympanic process, which develops as an extension from the first pharyngeal pouch. This will develop into the tympanic cavity and the auditory tube. Ossicle, these are the malleus, incus, and stapes. These are developed from the first and second pharyngeal arch. Malleus and incus that develop from the first pharyngeal arch, and the stapes that develop from the second pharyngeal arch. Then muscles related to the ossicles, these are the tensor tympani and the stapedius muscle, right? The tensor tympani, which is related to the malleus and incus, they are developed from the mesenchyme from the first pharyngeal arch, and the stapedius, which is related to the stapes muscle, is developed from the mesenchyme from the second pharyngeal arch. And the tympanic membrane, it consists of three layers. It's very, very important Viva question. Tympanic membrane, it consists of the three germ. It develops from the three germ layers: the ectoderm, mesoderm, and the endoderm. External auditory tube, it is formed from the first pharyngeal cleft. Auricle, it is formed from the 6 helix. The 6 helix, first, second, and third are from the first pharyngeal arch, and fourth, fifth, and sixth from the second pharyngeal arch. So by this, I'll end this session. Hopefully, you have understood about the development of the internal ear, middle ear, and external ear. Thank you.